and then hepatitis viruses, which is also pretty substantial. Are you guys tracking that I'm gonna take Major Johnson's slot on Wednesday? Okay. Just looking for reactions. Everyone okay with that? Not devastated? Okay, alright, cool. Yeah, I think the full complement of lectures for Block 4 is probably a good thing, so. As much as I hate lecturing right after exams, and you guys probably feel the same way, I'll try and just start Block 5s like on the scheduled day so I don't run into this problem again. But at least we're not adding an extra lecture to Block 4. Okay, so rotaviruses. Start off with these unusual viruses. They are unusual in that they have a triple layer X capsid. So they are capsid viruses, which, as we talked about for enteric viruses, is kind of de rigueur how they survive the low pH of the stomach. Rota means wheel, so if you look at an electron micrograph of these virus particles, you can kind of see that morphology where they have a couple different layers of capsid protecting that genome. And then the genome itself is also pretty unusual. It's in its own little classification of Rheoviridae, this family, which stands for respiratory, enteric, and orphan. So the orphan is a designation of the fact that it's the only virus in this family. It's a double-stranded RNA virus, very, very strange. It has a strange replication mechanism that we don't have to talk about, but it is kind of important to know that it is a segmented genome. So because of that, it has implications for the potential to resort. Non-segmented viruses don't have that potential, but viruses like orthomixed viruses, influenza, for example, and rotavirus, this thing can reassort. And that can create increased genetic diversity in this virus population. There are 7 different serogroups. We do have a vaccine, we have a couple vaccines, Rotarix and Rotatec. Those vaccines have to cover, especially group A, I don't know if they cover other serogroups, but this is the one that's mostly associated with outbreaks. So again, this MC, that's an abbreviation for most common, just to save some space. You will see that in other courses as well. It's the most common cause, important to note, severe epidemic diarrhea in children. Before we started vaccinating in the US, which I think we did around like 2006, we started introducing Rotarix or Rotatec to the US population. We had a lot of cases of rotavirus in the pediatric population in the US. It's still a problem in the developing world, even though there have been a lot of efforts by Gavi and other foundations to get this vaccine introduced into developing countries. But worldwide, certainly in places that are not vaccinated, the death toll due to rotavirus is up there. Some estimates put it as high as 1 million deaths per year, and those are amongst children who don't have the immune response developed yet, they don't have a mature immune response, they're not able to clear the virus. And how they ultimately end up succumbing to this viral infection is usually dehydration. The diarrhea is so severe that it causes dehydration. When people, and adults can get infected as well, but you tend to get milder infections. When people get infected with rotavirus infections, there's a range of symptomatic presentation. You can be subclinical, you can have mild diarrhea, or it can be extremely severe, but it's usually non-bloody. This is one of those viruses, or one of those entire pathogens that's not associated with bloody diarrhea, non-bloody diarrhea. In terms of transmission, fecal-oral transmission is the primary route. So we're talking about mostly contaminated water, oftentimes. Feces from somebody who's carrying rotavirus. It's a very short incubation period, less than 48 hours. For diagnosis, we can do PCRs. This is one of those targets on a multiplex PCR panel, like the BioFire GI panel, for example. This has that. And also, we have a lot of immunoassays and rapid diagnostic tests for rotavirus. When somebody is shedding rotavirus in their stool, they're shedding huge amounts, like 10 billion or 100 billion virus particles per gram of stool. Huge amounts of virus, so there's no problem picking that up on a rapid diagnostic test. Contrast that with norovirus, this other enteric virus that we're talking about. So this too is a capsid virus, it has that in common, but there's a couple important differences. This belongs to a group of positive science RNA viruses called Leeske's RNA. It has a couple of related viruses, like staphylovirus and astrovirus, and can also cause enteric disease. But norovirus is kind of the most significant one of these viruses. It is very common, sometimes colloquially called the stomach bug. Norwalk virus is an older nickname. This is kind of getting phased out of usage, but that's because of the initial outbreak occurring in Norwalk, I think, which is in Ohio, so in the 60s, so they just named it after that place. It too has fecal-oral transmission, so mostly contaminated food and water. We get outbreaks of norovirus that are associated with places like just crowded and congested places, camps, daycares, schools, very famously cruise ships, sometimes called the cruise ship virus. If you've ever been on a cruise ship, this is one of those primary things that the cruise line industry is concerned about, preventing transmission. Some of those big cruise ships, they might have thousands of occupants at those ships. And if you have an outbreak, that can reach a number of people. There's no vaccine, so all these people are susceptible. You can get reinfections commonly throughout life. This is an RNA virus, so it mutates a lot. There's certainly been efforts to find a vaccine, but to date we don't have a good vaccine for norovirus. So one of those takeaways is that this is kind of age indiscriminate, whereas rotavirus, you want to focus on being more of a pediatric virus, although you can get infections in older folks. In terms of the incubation period, this is also a pretty short incubation, shorter than you would find with like enteric bacteria, for example. As little as 12 hours to have sufficient viremia to have these symptoms, which are nausea, vomiting, diarrhea, NVD, sometimes see that abbreviated as. And then with a short incubation period, fortunately, the disease itself is usually not going to last a long period of time. Within 2 days, maybe 3 days, within 72 hours, you should have resolved these symptoms and be perfectly fine without the need for drugs. So, and I don't think there are any specific drugs for norovirus infections. Diagnosis is pretty much the same kind of methods that we would use for rotavirus. We have PCRs, it's the same target on the GI panel. Norovirus is also in there. And you're also shedding huge amounts of virus in your stool. So, easy to pick up on rapid diagnostic tests, but the implication is when you're shedding that much virus in stool, and we're talking about, not to be graphic, but like explosive diarrhea that can be aerosolized. It's one of those things where you just need to be by yourself for a couple days until that infection resolves. Very important. All right, here's a question I have that is a hard question, probably harder than when I would put on an exam, but let me see what you guys think of this question. Have a look. I mean, yeah. Sorry, yes, it's norovirus. But why?
Non-bloody.
Non-bloody, that's a good name. So what would that, who said non-bloody? Who said that? Okay, cool. Excellent. So what would that kind of roll out on this list?
Dysentery, inflammatory bowel, and ulcerative colitis.
Yeah, ulcerative colitis, right? That implies that the colon has ulcers in it, which is usually associated with some blood, even if it's occult in stool. So A and E would not really be consistent with this. Let's look at B, inflammatory bowel disease. So that can be a broad term. It can be a type of it can lead to ulcerative colitis, but an inflammatory disorder can have infectious and non-infectious etiologies. Not really the first choice for this particular vignette, so I would not look to that one to be correct. If they were just down between these two, let's say we're at a 50-50, what are your thoughts on this one about B. serious? Remember we talked about B. serious in block 30. What? Oh, okay, okay. I mean, you're not wrong.
They didn't deny Chinese food.
Okay, so the vignette is providing you of some hints that you've gotten used to looking for, which is pretty valid, right? I mean, you don't hear it. There's nothing in there that tells you what the source is. Could have been fried rice. Yeah, you can be serious. But yeah, Megan, what do you think? Yeah, okay, so the two forms of beef series that we talked about were which forms? The toxin mediated and then the legit infections of the bacteria. So if the bacteria are involved, that's going to involve an incubation period too, and time for them to divide and replicate, multiply. So that is usually a longer period of time. With more toxin, remember that window where you get symptomatic after exposure to that preformed toxin? Yeah, it can be as little as like 30 minutes after ingestion of that preformed toxin ready to go, all the way up to maybe 6 hours, 8 hours. So question, I mean, you could say 8 hours, I would be pushing it. 12 hours starts to get pretty meaty for a question. But certainly not 36 hours, that's long for the toxin. So the way this is worded is food poisoning, not an infection. So that implies it's the emetic toxin medium. And this window, 36 hours, is reasonable for toxin mediated feed.
Was it the infection the actual bacteria diarrhea?
So in the diarrheal presentation, there are toxins associated with that too that B series produces. But the incubation period, the presentation itself is more of the diarrheal presentation. And you were asking something about the presentation vomiting.
Well, because I remember the toxin mediated was vomiting and then the actual bacteria infection was that.
Yeah, it's just more like the emetic is more associated with vomiting as a predominant symptom, and that like the bacterial replication and infection you get in the gut with viable bacteria, B. cereus, is more like associated with that diarrheal presentation. Yeah, but there's still toxin production even with that diarrheal form as well. Yeah, that's kind of, this has and diarrhea, so that, that vomiting might point you a little bit towards norovirus as well. That's sort of the thought process with this question. Just reading in depth into things like the incubation period and how long it lasts. I guess the incubation period is not here, but how long it lasts, and the fact that there's no fever and no blood in the stool. Okay, so those are the respiratory viruses. Any questions on rotavirus or norovirus? Okay, oh, one quick note about norovirus real quick. Just talking about the epidemiology and the association with, cruise ships. This has also been associated with some major outbreaks that are associated with shellfish too. We've talked about this with Vibrios shellfish being concentrated in those filtering shellfish. There was whole large outbreaks associated with norovirus ingesting shellfish as well. So they're able to concentrate viruses too. Something to keep in mind there. Okay, moving on to HIV. So HIV is the only retrovirus we're going to talk about in this course. We won't go into, well, we will talk about the replication because it's important to know how this virus replicates in order to understand some of the major antiretroviral drugs treating HIV. So we'll just cover that in a couple slides. It is, as a retrovirus, it's enveloped, medium-sized. We think that it probably has zoonotic origins. It's probably circulating in non-human primate populations, chimps, gorillas, and then it made a species jump from those species into humans and adapted to humans. Various sources will give you different dates on when that species jump happened, but it became adapted to humans, and now HIV, like there was a simian immunodeficiency virus, and now this particular virus is definitely adapted to humans, so it's very good at infecting humans. Human immunodeficiency virus. When it first started to make its way on the scene in the US, it was 1980s, early 80s, maybe late 70s, when we saw clusters of cases that were associated with distinct demographic populations. So we saw really rare, unusual diseases like Kaposi sarcoma, which we talked about before, which is caused by which human herpes virus? Like 1 through 8, which one? 8, yeah, HHV8, Kaposi sarcoma virus. So we started seeing like unusual, rare diseases like that pop up, and pneumocystis uribexi, which causes this unusual pneumonia that most people are entirely able to combat because we have functional immune systems. We saw cases like that pop up. Epidemiologists, more virologists started researching this, and they started characterizing HIV. And it became a very alarming disease very quickly as we realized how many people were getting infected. So we characterized transmission routes. We started educating the population on how it's transmitted. So, I mean, when I was a kid in the 90s, I remember basically being taught about how HIV is transmitted ad nauseum, and they were beating that horse dead because we didn't have antiretroviral drugs at the time, and we had no other way to stop or contain this disease. And also at the time, because we didn't have ART and the antiretroviral therapies, ART, it was considered to be a death sentence. So it was taken very, very seriously. Kids, don't use drugs, intravenous drugs, needle transmission, parenteral transmission, blood products, and then definitely specifically sexual transmission, which still, as you can see on this pie chart, still is like the predominant way this virus is transmitted. So we have a couple different forms of sexual transmission. Male to male sexual contact, MSM, managing sex with men, is still the number one route of sexual transmission. Entirely possible to transmit male and female, or each way, but it still comprises a relatively small percentage of those cases. In terms of transmission, we'll talk about this a little bit more. It's not the most efficiently transmitted virus, but with those populations, we got a huge number of cases in a relatively short period of time, especially when people weren't aware of how it was transmitted in those days. In terms of the global picture, this is 2022 data, but I don't think it's changed radically in the last 3 years. We're looking at basically 2 different types of HIV, HIV-1 and HIV-2. Worldwide, the dominant type of HIV is HIV-1 now. HIV-2 is still circulating, but that's mostly out in West African countries where you get HIV-2. And it is generally a little bit less virulent than HIV-1. In terms of the global picture of the number of cases and the incidence of HIV, there's some striking figures here that kind of speak for themselves. So one, the top 2 figures, we see that the current number of infections that we have, well not current, but 2022 data, 39 million people infected with HIV, with 630,000 global deaths attributed to HIV-related illnesses that year. So that is whatever percentage, like 0.4, 0.5% of those cases that are resulting in death. If you contrast that to the total number of infections since the late 70s, the total number of deaths since the late 70s, we're looking at a denominator of 86 million cases ever, documented cases, and 40 million deaths. That's a lot of deaths. It's one of the major infectious disease killers. So that's like 45% total of those cases resulting in death. What does that tell you if the total number of cases has resulted in about 45% deaths versus the number of deaths resulting from cases in 2022? Yes, I was sorry, that was a terribly worded question, but I think you guys got it. That tells you that these days in 2022, ART is working very, very well. We have prevention strategies, but more than just the drugs, we've also successfully managed to educate people about how it's transmitted. We've been able to talk to mothers about mother-to-child transmission worldwide. So programs like PEPFAR, if you've ever heard of PEPFAR before, that is a global program that is going in particular areas where there's a high prevalence of HIV, like Sub-Saharan Africa, South Africa, Lesotho, Eswatini, these countries, and making sure that people have access to ART and bringing those mortality rates down. And even more importantly, limiting transmission. All right, talking about the virology of HIV a little bit, with this particular virus. Important to know that its receptor is this glycoprotein called G120. That's the 120 is how much it weighs in kilodaltons, but that is on the outside of this particle. It's the primary glycoprotein that's responsible for docking to its host cell receptor, which is CD4, primary receptor. Also, we have the GP41 that is associated with that glycoprotein. That's the transmembrane portion of that particle. When you have that primary docking to the host cell receptor, GP41 will GP120 will undergo a conformational change. It will expose GP41, and that is what's responsible for fusing those membranes together at the plasma membrane. So GP41 has fusogenic properties. Moving inwards, you have this capsid or core that sometimes looks like a candy corn morphology. That's a weird morphology. That capsid protein is called P24. It's an important marker for diagnostics, and there's a lot of that in this virus. Yes, sir.
Well, the slide under GP41 says facilitates viral century facilitates fusion, correct?
Yeah, you're right. So spacebar didn't go far enough. The word fusion is obscure. If you want to just make a note that that's what it's doing. I might have that somewhere else too, but thanks for pointing that out. So it facilitates fusion between viral and host membranes.
So that's the penetration at that point, correct?
Yeah, exactly.
So 120 is the ligand, basically the lighter binding to handshake, if you will.
Yep.
And then that is supposed to be on.
That is correct. Yeah.
Just, I know, there's no cure, for the previous slide, but there is treatment. I thought for like target, I thought it was like really expensive and hard to acquire to actually get to
You're talking about a specific antiretroviral drug?
Yeah.
What's it called?
I thought it was like Barbie or something like that.
Hold that. We have a couple slides about antiretroviral drugs. See if you can recognize it on that list. That's not ringing a bell to me, but yeah, I definitely want to talk about some antiretroviral drugs specifically, as well as some other treatment approaches that are interesting. So yeah, hold that thought. Okay, so P24 important when we're diagnosing it because we can see this protein on Western blot, so we can use this as an indicator that this virus is present. And then inside the genome itself, these are two positive sense RNA strands. They are identical to each other, and that has some implications in terms of like its ability to undergo some limited reassortment if you have a co-infected cell. But two copies RNA genome that have to be reverse transcribed in this viral replication process. It does have a bunch, being a retrovirus, it has to be able to do a bunch of things in order to integrate into the host genome, so it carries some accessory enzymes along with it, as in integrase, for example, some proteases that allow it to cleave its own products, but those are all maintained in that viral capsid core. All right, a little bit more detail about how this virus binds to those cell receptors. So if its primary receptor is CD4, you guys remember what type of lymphocyte that's expressed on in large numbers? I mean, a recursive question, but CD4 T cells, lymphocytes. So those are your T helper cells, right? They express a lot of CD4 on their surface. They're not the only cells, not the only immune cells that express CD4. Macrophages also will express a little bit of CD4. So this chart is showing you a CD4 cell. This like snowman-looking blob right there is CD4. It's depicting that you have CD4 on the surface. The virus is coming into contact with CD4, and on that virus surface again you have GP120 as that glycoprotein that's making that initial interaction with CD4. This virus, in order for it to efficiently infect cells, it requires a co-receptor. So it can use a couple different co-receptors in order to gain entry. These are chemokine receptors. There are two major ones, CCR5 sometimes called just R5, and then CXCR4 or R4 or X4. So those receptors are needed in order for this virus to be down to the plasma membrane, and then for GP41 to do its thing and fuse with that membrane. And there you go. Binding triggers fusion between GP41 and the cell membrane, allowing viral entry. A couple things about those chemokine receptors that are important to mention here. There's more of an association with CCR5 and macrophages, and this other kind of receptor, CXCR4, with T cells. So that has some important implications as well. Just keep that in the back of mind. So we've entered the host cell. We're in the cytoplasm now. We've uncoated. The membrane is already fused. We have our positive RNA genomes now in the cytoplasm. They have to be reverse transcribed, so there's nothing in the host cell that can do that function. That means the virus has to bring its own polymerase, which is called a reverse transcriptase. These reverse transcriptases are extremely replication, like high error rates, very low fidelity enzymes. So they make a lot of mistakes when they're synthesizing DNA copies from RNA. But that's what they do. Reverse transcriptases will have to synthesize cDNA from that template. That cDNA has to be imported into the nucleus. Once it gets into the nucleus, then that virus has enzymes like integrases that allow it to integrate into the host cell genome. When the genome is integrated into the host cell genome, it's called a provirus. And it can stay there very stably for long periods of time, where host cell transcription machines recognize it like any other gene, and they start transcribing viral proteins or viral transcripts off of those viral genes. Those proteins, once they're made, basically just act like any other virus. They start assembling. You get copies of the RNA genome made, and you have regular virus assembly occurring at the plasma membrane. So they bud off, and then you can go infect new cells. That's kind of like the active cycle for HIV virus. But by virtue of the fact that you can integrate into host DNA and stay there stably, that's what makes this a latent virus. So you can either actively transcribe those genes, or you can quietly stay in that host genome and not transcribe genes and just stay there for years. This is why HIV infections have that latent period where there's years where you hardly have any symptoms, you don't have a lot of virus production, but it's because of that fact that they can stably integrate into the genome. The high error rates do lead to a large population of genetically diverse virus particles. So in RNA virus, you have a ton of different errors that accumulate, and because of that, you get these quasi-species. The quasi-species of HIV are basically like a bank of viruses that numerically can respond to selective pressures in the host. So if the immune system starts destroying those viruses, it might be able to destroy the vast majority, but the majority that survives can be selective. That's kind of the advantage that viruses have. They have these quasi-species like that. We're talking about many, many different types of quasi-species that can be in the similar look, like the cytosine. So that has implications for your ability to clear it from your own immune system through these infections. Infection of new cells, they start generating different types of viruses. These viruses might have more tropism for T cells, macrophages, and you start switching the target cells. And over time, that gives you a population that's able to persist in that host for a long period of time and survive in that host. This slide basically summarizes what we just talked through in the last couple slides with the pre-entry steps to docking to the primary receptors and the co-receptors and the fusion of the membrane, the entry and the uncoating that happens, the reverse transcription that happens out in the cytoplasm, and then the fact that these DNA strands have to be transported into the nucleus and stably integrated into the nucleus. But from there, what happens depends on whether or not you have active transcription or not. That has bearing on the course of the disease. When you do have active transcription, That's when you have productive production of virus particles. And when these stay quietly in the genome, you have no production of virus particles. Okay, so in terms of significance, let's see. Hang on 1 second, sorry. Okay, so just getting my bearings here. So when you have an exposure to this virus, let's say your transmission is like sexual transmission. The virus in, it will be initially attacking and entering mostly macrophages in the genital area mucosa. Those macrophages will, along with dendritic cells, take up this virus and move it into regional lymph nodes. And in those regional lymph nodes, if there are viruses in that population that can also infect T cells, like those R4 viruses, then you can get infection of T cells as well. But early on in infection, during the initial and acute infection, this typically lasts between 2 to 4 weeks by the innate immune cells, so macrophages and dendritic cells. Once your adaptive immune system kicks in, that's generally when you've had that virus taken into lymph nodes, processed, presented, and you've had T cells that have started to become activated that will recognize HIV antigens. So you'll start getting a specific anti-HIV response at that point, after a couple weeks. And that's when you can start getting some of these initial symptoms that are due to acute HIV infection, when a person might start realizing that there's something wrong with them. It will have a fever, chills, malaise, like the typical flu-like symptoms. This usually corresponds to that 2 to 4 week period of early acute infection. Here, high levels of capsid protein are detectable. So that capsid protein that I was talking about, diagnostically is very useful early on in infection. When you start having initial high levels of viremia, do immunoassays like Western blots, for example, and look for that P24 antigen. You can also look for exposure, so you can look for antibodies to HIV. Those can be variable, occurring anywhere between 1 all the way up to 10 weeks. So early on in the infection, you might not see any antibodies, but eventually you'll start seroconverting, and you'll see antibodies against HIV antigens as well. As those lymph nodes, as the virus starts replicating in lymphocytes in lymph nodes, that can spread throughout the body, and you can have reservoirs of lymph nodes that have high numbers of virus in them. So it's a stable population of HIV that's very, very difficult to get rid of and can persist for long periods of time. So the wait period at this point, initially it's better seen in the chart next slide, but once your adaptive immune system kicks in, you will have a significant impact on the viral populations in the host. So your T cells will be activated, cytotoxic T cells will start destroying infected cells. Your immune system will basically do what it's designed to do, which is dramatically decrease the number of viruses in that person. For a while, I am just going to skip this slide and talk through on this slide. For a while, if you look at the viral load and how that's impacted by the adaptive immune system, it looks very promising for the patient. This data is showing what would typically happen if you don't have any kind of retroviral therapy for that patient, so no antiviral drugs, what naturally happens. It's a complicated graph, there's a lot of information here. On the left y-axis, this is the viral load, and that's in that red dashed line here, so you can look at that over time. On the right y-axis, we see CD4 cells, T cells over time, what happens to them. And then here we have the progression of disease from acute phase to latent phase, where we start counting time in terms of like months or years instead of weeks. And then finally, what happens when you progress to fulminant AIDS. So early on during the acute phase, like I said, you have activation of the adaptive immune response. If you just look at the viral load, it's going to peak, and that's usually when the virus is successfully replicating in lymph nodes and infecting those immune cells, right? But when your adaptive immunity kicks in, you get that under control, you have your CD8 cytotoxic T cells, and you also have antibodies. Antibodies are important for clearing those HIV particles with this infection. So between the humoral response and the cellular response, you do a really great job at eradicating a lot of those viruses early on in disease. That's sort of like the end of the acute phase at that point. And it looks like you would have succeeded in one and cleared the virus, but over time what happens again is those viruses go quiet in your genome. And if there's nothing being expressed, there's nothing to kill for the T cells and for the B cells, for the antibodies. So it just stays that way. And then slowly, very, very slowly, you have a little bit of transcription, a little bit of viral replication, and your CD4 counts start decreasing over time. Meanwhile, your viral load starts increasing, and eventually, around this point in time, when your CD4 count drops below 200 cells per microliter, that's usually where you were no longer immunologically capable of fighting out basic pathogens. You become susceptible to a wide number of diseases. So this is kind of when things start getting really, really bad. Very hard to treat at that point in time. Basically, just have to make sure somebody doesn't get exposed to those diseases because they don't have the ability to fight them off anymore. And in terms of the HIV antibody levels, those will peak to right around the end of the acute phase and slowly diminish over time. So looking at opportunistic infections and what this means in the lens of your CD4 counts. So if you start with a CD4 count of around 1,000 T cells per microliter, and over time you start looking at what happens when those drop down to clinical definition of AIDS, around 200 here. Some of the first infections that you become susceptible to occur when your cell counts are still well above that 200 threshold, 200 cells per microliter threshold. And you might start getting some bacterial skin infections you wouldn't normally get. Maybe even some fungal infections like athlete's foot that we'll talk about in a couple days. Most healthy people don't get athlete's foot, at least not sustained athlete's foot. So you start seeing slow, subtle indications that this person's immune system is shot. A little bit further along, you become susceptible to things like shingles. So whereas a normal person wouldn't have necessarily an issue with reactivated virus, once your T cells drop below, let's say 400 or so, 600, 300 maybe, reactivation of varicella zoster virus might happen at that point. Shingles, a little bit lower, you get thrush, which we'll also talk about. That's a species. You don't see thrush in immunologically competent people, and so on and so forth. Once you get to this point where you have a red dashed line here, these are all AIDS-defining illnesses that you do not see in healthy people. Okay. So we'll talk about, if we haven't talked about these, we will continue to talk about a few of these, like pneumocystis we'll still talk about, Cryptococcus, Cryptosporidium, but we've talked about the MAC complex. We talked about some of these already. So. Okay. Let's see how we are doing on time. Let's go ahead and take a 10-minute break now, and then we'll come back and talk about art. And the efficacy rates are, what it's designed to do is to basically, prevent you from contracting HIV, high risk things, so. Or if you're in a relationship, is statistically transmitted. But is there no risk? Going back to this slide about basic infections, I was completely remiss in not drawing some attention to tuberculosis. I spent a huge chunk of time talking about tuberculosis and its relationship with HIV. There's a lot of different combinations of problems that can happen when you have co-infections with tuberculosis and HIV. But on this particular chart, what I kind of suspect they're indicating is that if you have like a high prevalence country where you have lots of TB and lots of HIV, high prevalence of HIV, high percentage of the population that is carrying latent tuberculosis. If you're a latent TBI person and you have maybe granulomas or whatever, and you become infected with HIV, what happens, do you think, as your C4 counts start dropping off? What happens to those granulomas that are containing your tuberculosis cells? Yeah, they kind of give out. So then you have reactivation cases of tuberculosis. Again, like you wouldn't normally get. So I just wanted to underscore that point, that relationship. We'll check a little more. Okay, so talking about HAART, antiretroviral therapy, formerly known as HAART, highly reactive antiretroviral therapy. These are the drugs that we have evolved over the last couple of decades, spent a lot of R&D funding to improve the quality of the drugs, diminish the number of side effects, all with the kind of specific goals on this slide in mind. So when we're talking about the purpose of ART, it's not with any intention that we're going to be able to eradicate the virus. Once that virus has established latency in T cells, there's not much you can do to coax it out. Although I will talk about a couple of development strategies or emerging strategies where we can do that. But with ART therapy, these are the goals. So not to cure the disease, but you want to keep that level of anemia low, like below the level of detection. And there are a bunch of related implications there. That should keep your CD4 counts high enough to protect you from opportunistic infections. Should keep you alive a long time, so you have a pretty normal lifespan at that point. Reduce HIV-associated morbidity, and then, very importantly, reduce risk of transmission of HIV to other people. HIV is already a virus that may not necessarily transmit 100% of the time, so you don't want to tell people that because it makes it seem like you can be risky with behavior, but just because you have with somebody, for example, who has HIV doesn't guarantee you're going to get the virus. There are a lot of factors that play those is viral load. So if you have a very low viral load, the risk of transmission really drops, and that's one of the goals of ART, is to keep viral load down so you're not transmitting to other people. Going back to other transmission mechanisms, blood supply. Before we well characterized the virus, we didn't know to screen our blood supply for viruses like HIV and other bloodborne pathogens that kept vaccine. Now we do, so now most of our recipients for blood transfusions do not get HIV through that mechanism because we so thoroughly screen blood for that virus. In terms of the length of time you need to stay on ART, this is considered a lifelong therapy. Fortunately though, nowadays you can get most of your ART from a single pill. They formulate multiple drugs, so there's at least 2 different antiretrovirals, usually 3, formulated in one pill, which does a lot for patient compliance because this is going to be this is going to have to be a lifelong thing. When patients stop taking their ART pills, that causes a lot of problems. Immediately, the virus will start reactivating, and you'll get increase in viremia, but you also get selection for viruses that have some resistance against those drugs. So that's a big problem in making sure that you'll maintain that treatment for the duration of your life. There is no bona fide vaccine for HIV, even though there's efforts to develop vaccines. But there are some really effective treatments, prophylactic treatments, that will reduce the chance of transmission even more. If you've heard of PrEP before, that is a therapy that has two, like a dual combo therapy. Tenofovir is one antiretroviral and then another one that I can't remember. But with PrEP therapy, this is usually suggested or recommended for people who are either engaging in high-risk behavior, like sleeping with multiple people and not wearing condoms, things like that, or HIV discordant couples where one is HIV positive, the other is HIV negative, and want to keep that negative person negative. Taking PrEP will reduce the chance of transmission by like a 97% reduction in the chance of transmission of this virus. So that's been really impactful as well. In terms of cures, there have been a couple really high-profile cases of people who managed to be functionally cured of HIV, which means that even without ART, they don't have any active replicating virus. That's a functional cure. And that's through usually some really rare genetic factors. There's a couple cases I'll mention in just a second. And then to contrast that, there's sterilizing cures, which means you've completely eradicated the virus. There's no difference in terms of the outcome. A functional cure and a sterilizing cure. Just sterilizing cure is probably not realistic for most intents and purposes. For the people who've managed to eradicate the virus, those rare instances have involved people who have cancers like acute myeloid leukemias, or another person who had a lymphoma. These are famous cases, the London patient and the Berlin patient. These people, because they had chemotherapy basically to irradiate cancer cells in their bone marrow, they had to receive stem cell transplants. And the stem cells that they received just happened to come from donors who had a rare genetic autosomal recessive trait where their CCR5 was deficient. It's called a delta 32 mutation receptor. So the cells that they received no longer have the CCR5 receptor, and that made it impossible for HIV to continue replicating in those people. And so they not only got a stem cell transplant from donors with this delta 35 or delta 32 mutation, they not only had treatment for their cancer, they also had a functional cure for their HIV at the same time. But very rare, not scalable. We've definitely looked at it for inspiration for treatments and cures, but it's not scalable, it's not reproducible, unfortunately. And there have been other kind of pretty radical strategies as well, like using CRISPR-Cas9 to edit halogen genes from embryos, basically. you guys know what CRISPR is? Gene editing a little bit, heard a little bit about it. So that's when you can use like RNA to guide endonuclease basically and do editing of entire genes and sections of genes. You can't do that with somatic cells in a person who's already, alive. You can go into an embryo before it starts dividing, and you can edit out non-functional genes or make mutations. So that was actually done in China in 2018 on fertilized embryo with it was from an HIV disordering couple. The father was HIV positive, the mother was HIV negative. And this researcher, who I think was prosecuted for doing this, he used CRISPR-Cas to edit out that part of CCR5. So essentially, the twins that were born are refractile to HIV infections for their life, for the rest of their life. And that was widely condemned across the international community. Tony Fauci came up on the net and said that was a terrible idea, it's not necessary to do that. So they definitely discourage gene editing for things, even for noble purposes like lifelong immunity to HIV, because we have such good treatments, because we know about preventing transmission, because of mother-to-child transmission, we can prevent that, a whole bunch of reasons. But the intent was to show that it is possible. And those two girls who are now, like 7 years old or so, are apparently, those two twin girls are apparently fine, but there's a lot of risks to doing that as well that people are concerned about. So that's kind of what those radical therapies that has actually been done. And then other strategies that are less radical, that are being researched at places like UCSF and UNC Chapel Hill. A lot of labs are working on different methods to cure HIV functionally, maybe not sterilized cures, but one of them is the Lock and Lock method, where these latent viruses are basically frozen in the genome, so they can never reactivate. Another is the kick and kill method, where they use anti-latency drugs to coax those viruses into expression so they start transcribing. That's super high risk though, because once you coax them out of latency, that means that you're putting all your eggs in one basket and you're relying on the immune system to clear that virus completely. So, strategies, none have gone to clinical trials yet, but those give us some hope for functional cures that can be scalable to the population at large. All right, in terms of targets, yes.
Question: is there any implications, I guess, that can come from deleting the CCR5 co-receptor?
Oh, that's a great question. So, yes, yes and no. The thing about chemokine receptor is when you look at like cells that are CCR5, which again are mostly like macrophages, there's a lot of redundancy for chemokine receptors. So if you have a null mutation in one, even if you're homozygous mutant for CCR5, you have compensatory mechanisms where you still have chemokines that bind to other receptors. So there's minimal impact on your ability to, function if you have that CCR5 32 mutation. And a lot of people are heterozygous carriers for that mutation as well. So it's pretty rare, but the population that has the highest incidence of carriage for that is Northern Europeans. That's like 10 to 14% of the population that has that mutation. There's no functional or phenotypic consequence for carrying that. It's selected for either during the Great Plague or maybe during the smallpox epidemics, and it might have conferred some protection for for those people. Yeah, good question. Okay, so with these different ARC targets, you definitely do not need to memorize all these different types of drugs, but what you do need to have an appreciation for is that when we're talking about these antiretroviral therapies, you do need to have combinations of these. Those combinations need to occur in different classes so that you minimize the chances of resistance. So, for example, some of the mainstays are some of these nucleoside reverse transcription inhibitors. NRTIs, those are oftentimes used in conjunction with things like protease inhibitors, fusion inhibitors like Maraviroc, that's one of the old mainstays of retroviral therapy. As a CCR5, Maraviroc. There are fusion inhibitors, integrase, strand transfer inhibitors, a whole bunch of different classes of these drugs that target different aspects of the HIV life cycle. This is kind of an explanation of what some of these do. And you might take these next semester in farm. You don't need to memorize these for this particular course. Just again, be familiar with the fact that we have different classes based on these different aspects of the viral infection cycle, replication cycle. Any questions on ART or what its goals are, or anything that we covered since we came back from break? There, you had a question yesterday about, what were we talking about?
The vaccines,
Yeah, we're talking about vaccines for, chickenpox and shingles. Yeah, so the Varivax vaccine and the Shingrix vaccine for shingles. I looked into that a little bit, and really quick before I forget and move on, since that was a really good question. The purpose of Varivax, which is a live attenuated vaccine, is to, since it is a live attenuated vaccine, it does get in and infect cells, but you do both humoral activation and you get cell-mediated activation. So if you get primary exposure, you get any cell, usually they're much milder. And then, say you get exposure when you're 5 years old, and you have a normal, very mild chickenpox presentation. Fast forward 45 years, you're now 50 years old, your antibody titers are diminishing, your memory T cell titers are diminishing. At that age, you're eligible for Shingrix, and with Shingrix, it's a subunit vaccine, so I forget which one of the viral glycoproteins it immunizes against, but that primes the body and allows you to kind of regenerate an immune response for that virus. So should reactivation occur, you have kind of like a refreshed immune response to that without a live virus doing it at that point. Yeah, so that could be like a birthday present you give to yourself when you hit 50. You'll get Shingrix. It's a two-dose vaccine, but still, yeah. Okay, so we'll start with on a few slides talking about the hepatitis viruses. A bunch of different viruses here. There are 5 that we're going to cover. They belong to different families. They have different mechanisms of replication. A lot of differences between these 5 hepatitis viruses. But the common thread is that they have tropism for hepatocytes, the liver cells in the parenchyma of the liver. In terms of the common symptoms of hepatitis, you have these dipteric symptoms, or symptoms that are reminiscent of jaundice. What happens when you have your liver being damaged is that it ceases to function as it normally should. One of the functions of the liver is to process bilirubin. And when it can no longer process bilirubin, feed it, mix it with bile, make sure that bile gets injected in the small intestine, aids in digestion, and then ultimately gets released in stool. You get symptoms that are associated with this when it ceases to have that function. The liver stops behaving as it normally should. So the bilirubin itself, if it's not going into stool, some of it gets processed by the kidneys, and the kidneys have to compensate for that. Because of that, urine starts taking on a darker color than it normally would. Similarly, because the bilirubin isn't being released, like it doesn't end up in stool, your stool become like this pale, pasty color that's like distinctive. And then lastly, the bilirubin that gets into your bloodstream that doesn't get filtered out by the liver can get into circulation, and that causes your skin to take on kind of like a yellow color, even though whites of your eyes can become yellow, like this guy in the photo there. So those are like the major dipteric symptoms: the yellowing of the skin, dark urine, light stools, and also itching of the skin is another facet of jaundice. That's when the toxins are building up in circulation, they get into the skin and cause this itchiness. For the viruses that cause this, we have a couple RNA viruses: hepatitis A virus, hepatitis C virus, and hepatitis E virus. These are all positive sense RNA viruses. The HDV, hepatitis D virus or Delta agent, that is a virus, but it is a very unusual virus, but it doesn't it's not capable of causing infections on its own. It requires hepatitis B in order to cause infections and contribute to them. And then hepatitis B is kind of a standout on this list because it's the only DNA virus. It's double-stranded DNA virus. But otherwise, in terms of their relatedness, there's no significant relatedness between these different viruses. Alright, so let's start talking about hepatitis A virus. This is transmitted fecal-oral route, and we have a vaccine for it. So the 25,000 cases in the U.S., that's probably on the high side, and now the vaccine that is routinely given to the population, these cases have significantly dropped. That said, we still get outbreaks of hepatitis A, and those outbreaks are usually related to contaminated food or water. A couple slides, I'll just sit there right now. These are some of the outbreaks that are typical for hepatitis A virus, with this virus getting its way into the food supply. There was a 2019 outbreak that happened associated with Woodman's, and they had a couple produce types that were infected with hepatitis A. That might have been because you had contaminated irrigation water or something, but the virus made its way onto this produce and resulted in a couple documented cases. A state outbreak in 7 states, I think, were implicated with hepatitis A. So the people who got sick were probably unvaccinated people who just happened to get last periods in time. Those were the implicated food sources from Woodman's. Other outbreaks can happen in places like restaurants. You can also get this from swimming in a swimming pool, where somebody is infected with HIV and it gets released into the water and can infect a lot of people that aren't vaccinated against it. You shed a significant amount of virus, and you shed it through stool, so because of that, it is possible to transmit it to the person. Sexual contact is possible as well, because you're shedding virus. Children are silent reservoirs because of the fact that, particularly like really young children who don't have developed immune systems yet, they don't have severe symptoms necessarily. They may be asymptomatic, but that's because they aren't putting the virus as effectively as adults are. So they're more susceptible to infection, right? For that reason, they're considered silent reservoirs for the disease. This is an enveloped virus. Oh, I'm sorry. This is a non-enveloped virus. It can remain 5 months outside the body, so that has implications for transmission as well. With food handlers, this is something you see oftentimes if you're going to the bathroom in a restaurant. There'll be a sign that says "wash your hands to reduce transmission," and then you'll see a bunch of foodborne diseases. Hepatitis A is usually listed on that list of foodborne diseases that are telling people, particularly in a setting where people are eating or preparing food, to watch out for. So a lot of cases are implicated with food handlers. In terms of the biology, again, capsid-virus, RNA genome here. The vaccine itself is a inactivated vaccine. You should get lifelong immunity for hepatitis A. There's a passive therapy is available as well, immunoglobulin is available for this disease. Looking at the pathology a little bit, this figure is out of the Murray textbook. It's a super unusual way of depicting the way this virus moves through the body. But whatever, it works. So if you go all around, you ingest it, it goes into your small intestines, it goes into the blood, and then ultimately its way into the liver, where it's replicating in the liver. So most of the replication will occur in hepatocytes, very slow replication. Small number of small intestine cells are susceptible to infection, so you get a little bit of viremia in the small intestines as well. Most of that is going to the liver. Where you shed it, again, you have some viremia in the liver, and that is a little bit of virus being mixed in with bile, which ultimately will end up in stool. So that's how it's transmitted and cycled and able to infect new people. One of the BHD is infectious for 2 to 6 weeks. I have to caution you with blood being infectious. It might be a little bit infectious, but there's not a lot of virus in blood. So the primary mode of transmission, remember, is fecal oral route, not parenteral. You want to associate that with other hepatitis viruses, but not with fecal oral primarily. And during this infectious period, that's symptomatic phase as well as the latter part of the incubation period. So you start shedding virus before you're even symptomatic with this infection. There's no chronicity, which means that this is a acute infection, you get sick, and then you resolve the infection. It doesn't have any reservoirs in the body where it can hide out indefinitely. Yes, sir. It's an indication of replication numbers. So high levels of replication occurring in the liver and then hepatocytes where it has tropism for those cells, and much less replication, but still some replication in the small intestines. Okay, hepatitis B. So this is one of those bloodborne pathogens. It is a significant virus. Nearly 300 million people chronically infected. Millions more acquired new infections. And note the infectivity of HIV relative to HIV, this other bloodborne virus we were talking about, is between 50 to 100 times more infectious than HIV. We'll talk a little bit about its association with liver cancer, but it is associated with HCC, hepatocellular carcinoma, and I think the third virus we've talked about in this course that it's oncogenic or linked to cancers. You guys remember the herpes viruses that were linked to cancers? Epstein-Barr virus, yep. And there was another one. The one that we talked about earlier. HHH8, which is sarcoma. So this too is to cancer, but the cancer specifically is a liver cancer. Leading cause of liver cancer globally. That's a big, huge deal. We do have a vaccine against hepatitis B, so that is really important. We vaccinate widely in the U.S. And there are hotspots for carriage globally, such as sub-Saharan Africa and East Asia. I don't know if there's a date on these stats, they're relatively recent on these cases. In terms of transmission, it's transmitted much in the way that HIV is, but it's more associated with contaminated blood and body products. So usually when you think about blood transmission or bloodborne transmission, you think about transfusions and things like that. But some of these, because it is so infectious, are things you gotta keep in mind. The old advice, you shouldn't share each other's toothbrushes and razors. That's really good advice for reasons like this. You can transmit viruses like HIV so efficiently with just minute amounts of contaminated or infected blood are sufficient for that. This virus, it can be shed in a number of different bodily fluids, everything from milk, vaginal secretion, semen, blood, and also saliva. But with like saliva, for example, even though you might be able to detect virus in saliva, the reason why you don't share toothbrushes is not because there's spit on the toothbrush, it's because there might be like this in your gum, where there's like minute amounts of blood. That's a far more efficient source of transmission for this virus. This figure, there's a lot of information in this figure, but it also kind of hints at some of the immunopathology of hepatitis B infections. So parenteral transmission, which virus gets into your system, into your bloodstream, makes its way towards the liver, and then very slow replication in the liver. Some of the significant pathology associated with hepatitis B is immunopathology. We haven't talked about it yet, but this virus produces an antigen called surface antigen, hepatitis B surface antigen, HBsAg, on that slide. And that antigen, once your adaptive immunity kicks in, you'll start producing antibodies and that will form complexes with the surface antigen. And that will stimulate some of this, classic immunopathology. It will cause inflammation of the liver, and they'll contribute to some of the symptoms that you associate with hepatitis infections, like right upper quadrant pain because of your inflamed liver, hepatomegaly, and enlarged liver, and some of those other symptoms. That's because of the immune response to the infection. You also get substantive good and bad, double-edged sword there. Without CMI, you don't get very painful symptoms or severe symptoms, and you also don't clear the virus in the first place. So it's kind of one of those no pain, no gain scenarios where you need that cell-mediated immunity in order to clear the virus. If the virus isn't cleared, you start shedding this in a bunch of different bodily fluids, and it contributes to this transmission cycle. But this year, maybe the immune response is what we're going to talk about next, for the next couple slides, and how important it is and how it determines whether or not you resolve the infection, and it's just an acute case of hepatitis, or whether you progress into a chronic disease state for this virus. Whether or not they're asymptomatic all the way up to a fulminant, potentially deadly form of hepatitis depends on your immune status as your immune system handles the infection. The incubation period is very long, minimum of 2 months, all the way up to 6 months or so. The symptoms associated with this acute phase are kind of mild flu-like symptoms. This wasting is characteristic, where you lose your appetite sometimes. You might have abdominal discomfort, other symptoms like that, nausea, aches and pains. The ecteric phase is when you would start to see you have a hepatitis virus specifically. And those can last for a month or 2. This part is kind of important as well. With chronic infections, I added a little bit to help specify what these stats are referring to. With children, again, because of the fact that children don't have a good, well-developed cell-mediated immune response, they end up shouldering a large percentage of cases that progress to chronic infections. So their immune cells are not able to clear those infected hepatocytes, and within 6 months, that's kind of the defining period where it becomes chronic hepatitis, acute hepatitis. Kids, 25% of all new HPV infections occur in your childhood, whereas most adults are able to avoid progressing to the chronic stage. Kids are not that lucky, so kids are the ones who typically progress to chronic HPV infections. With adults, only 5% progress to chronic infections. And a very small percentage of those will die from these potentially fatal outcomes like cirrhosis, where you have scarring of the liver and loss of liver function, so it fails to do what it's supposed to do. It's no longer filtering toxins, it's no longer processing bilirubin, or you get liver cancer. Our slide that kind of walks us through these different outcomes and how these different outcomes are determined. You get your exposure. 90% of people will clear the virus. So eradicate, no more virus, no chronic stage. The latent stage is completely cleared. Less than 10% will go into this chronic stage, which again is defined as having this virus in your system for over 6 months. And then a very small percentage will progress to fulminant hepatitis, which is usually fatal, rapid onset, liver failure, and very poor prognosis for those infections. For these chronic infections that last for a long time, where you have the surface antigen detectable in your patients for longer than 6 months, those guys, eventually, some of them will clear the virus and you'll get complete resolution in about half of them. Others will maintain themselves as a carrier state, so they're shedding the virus, but they're asymptomatic themselves. This is not something you want to let go because they are carrying the virus. They're potentially infectious and are asymptomatic. And then others will become chronic persistent hepatitis patients. These patients worry about some of these extrahepatic diseases or complications. So outside the liver, you can get polyarteritis nodosum, which is when medium-sized vessels start constricting, and you have potential for things like ischemia, infarctions, circulation problems that can arise from that. So it would be unrelated to liver function, but it's a complication that can happen from chronic hepatitis. Another potential complication is glomerulonephritis. We've talked about that before. We've talked about glomerulonephritis as a sequela or bacterial disease. Yeah, or strep throat. So we can see that again here too. In this particular sequela, your longer filter. So they're already compensating for liver failure at that point, and that puts extra pressure on your kidneys. If you have both glomerulonephritis and cirrhosis in the liver, that's a really bad clinical picture. And then this last category, we have chronic active hepatitis. This is where you can get cirrhosis and hepatic cell HCC. In terms of the virus structure, this is a pretty small virus, 42 nanometers, a tiny little guy. It's enveloped DNA virus. The most important thing to remember is the surface antigen. That's the virus protein. Is what allows it to survive out in the environment for such a long time, especially for an envelope virus. That's a very, very distribution of surface antigen on top of it. On the genome, the double-stranded genome, which is pretty small, 3,200 base pairs, it's a tiny genome. It has core antigens protecting it, nucleic acid, core antigen, and it also has this E antigen, which it can secrete outside of the virus particle. So you can actually raise antibodies not just against surface antigen, you can raise antibodies against E antigen as well. This particle is called a Danian particle. In terms of diagnostics, we need to cover this serology panel that's oftentimes ordered for hepatitis B and how you interpret it. We can use both the antigen and the antibody as markers for this serology panel that's ordered for hepatitis B. We can use this information to diagnose whether somebody has a current infection, whether they've had a past infection, acute or chronic, and whether or not they're actually shedding virus. We get a lot of information ordering these panels that are basically just looking for either the antigen from the virus or our own immune response to these different viral antigens. For these antigens, that surface antigen is the first marker that shows up when somebody is infected with HBV. So before you get antigen to any other, or before you raise antibodies to any other marker, this will be the first one that pops up. You'll also get antibodies eventually to E antigen, and that is interpreted that you have an actively replicating virus in the liver, and that is when people are pretty infectious at that point. So if you see E antigen on a serology panel, that usually indicates that person is infectious. With antibodies, E and antigen, hepatitis B infections, if that happens, you should no longer be able to detect the antigen and see those antibodies starting to pop up. So we'll look at a couple of examples and what that looks like. All right. So here he's showing us an acute infection. The patient is symptomatic. They have bacteric symptoms. They have those general flu-like symptoms. And if you were to order a serology, you should be able to detect that surface antigen and the E antigen. Again, the way you infect that, or the way you interpret that, is that surface antigen indicates that the virus is there, but that E antigen is telling you to eat something even more informative, it's infectious. You won't be able to see any antibodies until those antigens go away. They just indicate you clear the virus. When you clear the virus, you'll see those antibody titers increase, and then you should see a diminishment of the surface antigen. With chronic infections, you simply do not seroconvert. So you continue to detect antigen, but you will not see the antibody for those chronic infections. For lab diagnosis, we have a bunch of things we can look at. First of all, the picture itself is the patient presenting with bacteric symptoms. A history which indicates that they might have hepatitis B. Have they been vaccinated against it? That's all relevant information to assess. But we can also order the serology. We've talked about how to interpret it, and I have an example of how to interpret that also, or a chart from Murray textbook. If somebody is diagnosed with hepatitis B, whether it's acute or chronic, you can also monitor their viral load using PCR, quantitative PCR. So if you have an antivirals, that would be an important thing to monitor over time. For acute infections, because 90% of people do clear their virus within 6 months, 40 treatment and is usually considered adequate. But if people are still coming up positive on serology panels after 6 months, then they might have progressed to a chronic state, and then there's some specific antiviral meds that are available to treat those patients to prevent serious cirrhosis and liver cancer as well. Alright, this is, like I said, the Strong-Murray textbook. This is an example of how you would use antibodies and antigens from a serology panel to interpret where the patient is in terms of their infection and whether or not they're infectious at that point. Very quickly, kind of walking through this, if somebody, like I said, they've been vaccinated at some point in their life, but they've never been naturally infected with this virus, the only indication you should see on that serology panel is that they are positive for that surface antigen, because that's the only thing in that vaccine. That vaccine is a subunit vaccine, so it has the surface antigen, and that's it in it. If a person's never been vaccinated or exposed, this entire panel is blank. But everyone in this room, you've all been vaccinated against Hep A and Hep B, right? So based on the serology panel, most of you would have this result on the far right there. For the rest of this, it gets a little bit more involved, but the way they divided this is disease state on the left and healthy state on the right. So somebody has been infected and they've resolved the infection. The difference is you would now see antibodies against some of the real deal viral antigens that patient would be exposed to. So they have antibodies against the core antigen and then antibodies against E antigen at that point. Early on in disease state, I said that surface antigen was the first viral marker to show up. So that'll show up before any other viral antigen. You can see that pre-symptomatic. With the infectious virus, you also see a positive sign there. What do you think that is indicating for the infectious virus? Why is there a plus sign there? Like how did they determine that, I should say? PCR, probably. So PCR, they detected there's viral DNA and it's positive, and it remains positive until you resolve the infection. So that's a good marker to use to order a PCR in doubt. And the rest of this you can kind of study on your own, but I do want to just point out for chronic infections there, the only way you would be able to tell if this is a systemic infection rather than an acute infection really is time. If you've been able to test a patient over the course of months and they still have the same panel results, then they've probably progressed to chronic carrier state. Chronic infection state, I should say. Okay, a couple more viruses and then we'll be done for the day. The next one is also a parenteral transmitted virus, a bloodborne virus called hepatitis C virus. As an RNA virus, important to know, it has fairly high mutation rates. There's no vaccine available for this virus. So Hep A and Hep B, they do have vaccines. Hep C, no vaccine. Parenteral transmission like Hep B though. Okay. Also, in terms of the disease that can cause hepatitis C, you may get asymptomatic infections. This is associated with a much higher percentage of patients progressing to a chronic infection. So, remember, less than 10% of people infected with Hep B progress to breast infection. With Hep C, that percentage rate is between 75 to 85%. And that's because of the way this virus interacts with the immune system, and its control over the immune system is much natural killer cells. So you get these infections much more frequently than you do with Hep B. In terms of this stat, or this information about it being the most common MC, the most common co-infection with HIV positive patients in the US, that is due to the fact that it is transmitted through the same mechanisms. So if you're IV drug user and you contract HIV, you have a higher risk of contracting a virus, hepatitis C virus, for the same route of transmission. Same with sexual transmission, contaminated blood products, etc. But this virus is more associated with IV drug users than with hepatitis B. For that reason, it does have a higher prevalence in the prison population in the US. If you look at causes of death in the prison population due to infectious disease, guess what the number one cause of death is? It's like over HIV, over tuberculosis. diagnosis, it's this hepatitis C virus. A couple other things to point out here. When you have very severe hepatitis C infections, you can have accelerated progression to these potentially fatal outcomes like liver cancer and cirrhosis. Faster time course, insidious onset, that's important to know what that means. When you get hepatitis C exposure, you can be asymptomatic for months and then all of a sudden have onset of symptoms and have a very bad disease state. That's because this virus is replicating slowly in liver cells and not attracting a lot of immune attention. So it's considered like kind of like an insidious onset disease. Asymptomatic until symptomatic. It sounds like a paradox, but it sneaks up on people is the point. They're not aware that it has been slowly, silently destroying their liver cells for years without them having any idea that it's doing that. And in that meantime, since they are asymptomatic, people aren't going out and getting tested for HCV particularly, but they could be transmitting that virus to other people during that entire time period. In terms of testing, certainly for the blood supply, this is one of those viruses that we test in that blood supply for certain. If anybody's had exposure to HCV, you can see that in serology. They have antibodies against HCV. For HCV positive people, you can do qualitative PCR, you can do quantitative PCR as well as monitor drug therapies. We have some very expensive specific antivirals to treat Hep C infections, almost like scandalous, expensive drugs. It took acts of Congress to bring some of these prescription drug costs under control, but I saw specials on it, like on Dateline, I think, years ago, that it was costing tens of thousands of dollars for patients who had HCV to get medication for like a 12-week course of drugs and antivirals. So it took involvement from Congress, it took involvement from patient advocacy groups to pressure the pharmaceutical industry to bring down those costs, allow generic companies to start competing in that market. And then lastly, this last bullet is confirmed positive. It is important to have medical evaluation and counseling for those patients. They are particularly susceptible to things like alcohol consumption, where if you're not familiar or if you're not aware of the fact that you have an HCV infection and you're just quietly going about living your best life, enjoying alcohol all the time, you could be doing serious damage to your liver during that entire period of time. So that's one important piece of counseling advice I would give to a patient if they find out that they're HCV positive. Also, you'd want to make sure that they're counseled on being vaccinated for HPV and HBV as well, so they don't get co-infections, which are entirely possible if you get co-infections. Make sure they're vaccinated against those viruses. And then lastly, with HIV, you want to make sure that they are tested and screened for HIV as well. All right, penultimate virus for today is hepatitis virus. It's a satellite defective viroid thing. It's not capable of causing infection on its own. It requires co-infection with hepatitis B. It does have this little tiny little RNA genome, only 1,700 nucleotides, tiny genome. It codes for a couple proteins that are responsible for replication of that. But in order for it to bud from an infected liver cell, it has to use the surface antigen from hepatitis B. So without that virus, it just does not transmit, does not cause infection. If somebody either already has hepatitis B though, or you acquire it at the same time through the same route of transmission, like blood exposure or something like that, and you get a co-infection, this is when you get really serious accelerated fast track liver disease. So HPV and HEV together, you get more severe rapid onset disease, much worse prognosis, and just much more aggressive disease course in general. For diagnosing hepatitis D co-infections, we do have a test where we can look for whether or not people have raised antibodies against that delta antigen, hepatitis D antigen. And we also have PCRs that can look for markers in that tiny little 1,700 nucleotide genome that it has. For prevention, it is usually transmitted through the same amounts of HPV, like I said, but if you stop HPV transmission, as in you vaccinate someone, you ipso facto prevent HPV infections. Okay? So that's the best way of preventing HPV infections, just don't get HPV. Okay, and the last virus that we're talking about today, hepatitis E virus. This one is actually kind of easy to study, but in a lot of ways it's similar to hepatitis A virus. It's also oral transmission. It is another positive sense RNA virus. It's in a family called Capiviridae. Let's see. We don't have a vaccine for this virus either, unfortunately, but it's not super common in the United States. There are hotspots globally, usually in areas that have poorer sanitation. So when cases in the United States, those are oftentimes linked to travel to some of those hotspots. A lot of people carry this through contaminated water. As a zoonosis, you can get this virus from eating undercooked animal sources, and like food-producing animals, so pork, venison, boar, a weird association there, and the usual suspect, shellfish, also. In terms of the clinical course of the disease, it behaves a lot like hepatitis A in that it doesn't progress to chronicity, so not like hepatitis B and C where you get chronic infections, it's acute like hepatitis A. But notably different is that you do have higher mortality rates. And mortality rates due to hepatitis A virus are around 0.1 to 0.2%. These are about 10 times higher, still low, but 1 to 2% mortality rates. And you have disease that's significant enough to be fatal. Since it's acute, most immunocompetent people will recover fully. And this too, it can be diagnosed by the same methods that we talked about with other viruses. You can do serology panels in this as well as PCR, or PCR against the genome itself. Okay, that was a lot of information. Is there any questions? Yes, ma'am. Yeah, exactly. So I think this is like a zoonotic virus, where if you look like hepatitis A is more cultivated in the human population explicitly without animal reservoirs. But this is Capiviridae, probably able to infect by that way.
Is acetaminophen or not taking acetaminophen something you consider as a bad infection?
Not taking acetaminophen because of its.
Is it would you avoid that, or is that something that
I
you
don't know. For pain management, docs might have a way of managing pain for liver, out of concern for liver damage or contributing to the infection.
Yeah, just like an active hepatitis, or
Yeah, that was